Hello, everyone. It's Mr. Cudell. I hope you're doing well. I'm doing great because we are here. We're at the end of the first semester. We just got to get through this chapter, but some people think this is the best chapter of the entire semester. That's what I think, and the reason why is because all that other stuff is kind of like elemental biology. It's grade school stuff with a few new layers on top, but this is the chapter that changes every year. In fact, when your textbook was published, by the time it was published, it was obsolete when it came to chapters 18, 19, and 20. That's what I'll do when I'm with you in person, or however that looks. We will get together and I will get the latest and greatest stuff in front of you. There are some fundamental principles here that seemingly won't change, or they're kind of at the most basic level. So they're kind of the base that everything else is built upon. I love it. I think we've been working so hard to get to this moment. That's the way I look at this. I hope that you're in awe. Let this chapter wash over you. Ask a ton of questions that I won't be able to answer. And then, I don't know, we'll get you one day to a university where you can get all your questions answered. But please be amazed. I want you to ask yourself, if you were to take a skin cell, does it have the same DNA inside of it as a stomach cell? And the answer, of course, is yes. And so then the question becomes, why doesn't the skin cell create stomach tissue like the stomach cell can? They have the same DNA. They can make proteins. And the answer is because it's regulated. It's turned on and off on purpose at certain times. But the question becomes, how does this cell know not to make stomach tissue, but knows to make proteins that will help make skin stuff like keratin and these other proteins, collagen and all this other stuff that skin cells make? How does it know? I think it's a great mystery, but we know the basics of this, and we're going to go through that in this chapter. In a few minutes, we'll go over the lac and trypoperon. They'll tell you that it's specific to bacteria, and that's true and not true. We also do the trip and the lac operon in our bodies, but then we have many, many more layers of complexity than just that. So this is the most basic stuff, and I hope that you're amazed by it, and then I hope that you're like, wow, I can only imagine 50 times more complex than what we've learned in this class, what that looks like. Because it is happening in your cells, and we just don't know how all of it works. That's what biochemists do, as they try to figure it all out. Well, there's a couple other examples over the last few years that I've seen of things that are unregulated in bodies, meaning cells that have just been turned on and they're just creating tissues that they shouldn't be. In fact, I have a story. When my wife and I were in graduate school, we had a roommate, Nick. What's his name? Nate or Nick? And he was in law school at the University of New Mexico Law School. And he had a girlfriend. So his girlfriend was from Texas, and she would always come visit. And I was talking to her one day, and she's like, "I got to tell you what happened to my friend." I said, "What happened?" She said, "Well, she was sore, like her back was sore all the time." And she's like, "I don't know why that is." So she goes to the doctor. This is my roommate's girlfriend's friend. Yeah, okay, believe this story. So she says she goes to the doctor. They take an X-ray. They see something in there. Like, okay, we need to go in there and check it out. So they lay her on her belly and they cut her open in surgery. And the doctor looks inside there, and he pulls out a softball-sized mass of teeth and hair and nails. And he said that when he picked it up, the teeth started. No, I'm just kidding. The point is that there was a cell, obviously, that went that just got a message that says, "Hey," it went rogue, and it was like, "Hey, I'm going to start creating these other tissues. Maybe I'm not supposed to, but there's a message here that we got our connections crossed, and I'm just going to start making these proteins and making this stuff." So it's a pretty crazy story. I feel for the young lady, but as far as I knew, she recovered from the surgery. Well, how are genes regulated? And I want you to look at the board and go ahead and read this stuff that's up here. Each cell contains the exact same DNA, but only allows certain genes to be turned on at certain times. And this most often happens because of these things called repressor proteins, and they literally sit on the DNA at the beginnings of genes, and they say, don't turn this gene on in this cell. But you can imagine in a stomach cell, you do want to turn the gene on that makes stomach tissue. So this repressor protein pattern, and I kind of put it over there, there's DNA over there, and it has little green dots on it. Those are supposed to be these repressor proteins that are repressing the genes from being turned on. It looks different in every cell, and then as you can imagine, it looks different in every body. And what scientists are learning now, we'll get into this towards the end of this chapter, is that this it's called the epigenome. It's the markers and tags that sit on top of the DNA are actually more important than the DNA itself when it comes to creation of proteins and the health of individuals. I think it's fantastic because for so long people said, oh, DNA, that's what's going on. And we really know now that the DNA is a small part of the story. This is a much larger part of the story. So let's regulate some genes here. We're just going to look at number one. And again, this is supposed to be a bacterial model here. So go ahead and remind your neighbor what's happening here with transcription, please. Remember a little bit more of the detail here. Remind your neighbor what's going on here also, please. See, don't you feel smart? Remember, there's all these proteins that are sitting at the beginning of any gene, and they are the things that enable the RNA polymerase to jump on there and do transcription. Here's a picture I got off the internet. You can see how complicated it is, how many things have to be perfect at the right spot there. Seen this movie before, we're not going to watch it again. Just be overwhelmed by how much has to go on and how important it is that we regulate gene expression, because we can't let genes just start going, or else you can have some of those issues that I talked about and that we saw on the screen. Well, one of the ways that we're going to stop proteins from being created is through feedback inhibition. And we know this from early on, from like chapter 1, we talked about feedback inhibition. Remind your neighbor what's going on there and try to tell your neighbor what's happening on the right side of this picture. Go ahead and press pause. There's feedback inhibition, but the new wrinkle here is gene regulation. We can actually physically turn off genes, and sometimes you can see here there's a protein product, or this is an amino acid. It's a product down at the end of this pathway, and it's going to feed back, and literally often it will stick onto the DNA into like grooves between the nucleotide letters. It will fit perfectly there, and it will sit there, and it will stop its own expression. Or the expression of one of the enzymes that creates it, which I think is fascinating. Well, let's talk about bacteria. Bacteria often respond to environmental change by regulating transcription. So let's imagine that you have E. coli bacteria in your large intestine. Now, these guys, they cohabitate with us. It's a symbiotic relationship. They help our immune system. They help us digest food. They produce vitamins. They're super important. There's a bunch of different types of E. coli, and you have one type, and if you get a different type in your digestive tract, it can make you really sick. But the type that you have is great for you. And when you look in the toilet after you go to the bathroom, I read about 60 to 65% of poop is actually dead E. coli bacteria. And that's great. And then you'll just get more from what you eat, or they'll replicate in there. I think, some people like to have kombucha. I like to eat a few spoonfuls of yogurt every day. Some people take probiotics. It's just to keep your bacterial levels at a healthy level for you, and they help you with all these things so that you don't have an upset stomach. Well, natural selection is favored bacteria that produce only the products that are needed by that cell, meaning they don't make stuff that they don't need to, and they're super efficient when they do this. The way they do this is that they have genes, and this is called an operon model. And you'll see up here, there's the lac and the trip operon. And operon just means an operator-controlled model of how they regulate gene expression. A cluster of functionally related genes can be under coordinated control by a single repressor or a single on-off switch. We know what the promoter region is of DNA that exists at the beginning of a gene. But within that promoter region, there might be this thing called an operator. And again, all this is DNA. So the operator is just a sequence of letters that probably that's where a product comes and represses its own production, or these little green dots that I drew over here. These things are sitting on the DNA. And literally, because it's an A, T, C, or G, or whatever sequence it is, that repressor protein is shaped so that it will literally fit on top of those. If it was a different sequence of nucleotides, it wouldn't fit on there. It's amazing to me that not only the nucleotides confer some sort of information to eventually make a protein, but their very shape, because remember those nucleotides are all different. The A and the T and the C and G have different shapes. Those shapes actually are there, and through evolution, things have evolved to bind onto particular sequences of those shapes, as well as the information they confer downstream to make protein. I think it's fascinating to me. So the operator is just this area. Here's a promoter. Maybe there's some letters that exist in promoter regions down there. Within that, there must be a sequence of letters, maybe a few letters that we would call an operator, where a repressor would bind. Well, the operand can be switched on or off by a protein repressor. Let's see what that looks like here. It's this little red thing. And you can see shape is important here. You can see on the left side that the repressor is being made, and then once something binds to the repressor, it changes the repressor shape, and then it's able to bind to the DNA and repress gene expression. This might be a little video of what the repressor looks like that fits onto the operator region of the DNA at the beginning of a gene. Well, remember, if the repressor is active, meaning its shape is changed by something binding to it, then it binds to the promoter region, which has the operator in it. And then that little white thing in the top right corner of the picture is the RNA polymerase. You can see that no transcription is occurring here. No RNA is made. The repressor prevents gene transcription by binding to the operator and blocking RNA polymerase. Cool is that the repressor itself is a protein that was made usually upstream of the gene in which it controls, meaning thousands of nucleotides before you get to the gene. If you're going in one direction along the DNA, there's a gene there that makes a protein that we call a repressor. And what's cool is that thing then has a job, and its job is to go down there and latch onto one gene and either allow it to be turned on or off. So you can see how important and coordinated all this is. I'll give you this question from the last chapter. Why might these mean trouble for transcription? If you have inversion or deletion or whatever, you can imagine then that those protein repressors aren't made as efficiently as they once were, and then you can't repress genes from being turned on, or maybe they're turned on too tightly now because there was a duplication of the repressor or whatever, and obviously that's going to cause trouble. Well, the repressor can be in an active or inactive form. This is the little red thing. Depending on the presence of other molecules, you can see there that it's in an inactive form and transcription is probably taking place. The little thing that's going to bind to it is called a co-repressor. We've seen it as this little orange thing. As a result of an inactive repressor being there and it's not latched on, genes are going to be expressed, protein's going to be made. But if the co-repressor binds to the repressor, you can see then it changes shape so that it fits into the grooves of the nucleotides in the operator region of the promoter of the gene that it's trying to repress or control. Active repressor has a co-repressor bound. There it is. Gene's not expressed. So let's get to an example. Okay, so let's imagine that you have bacteria in your large intestine, and they need amino acids in order to make proteins inside of their single-celled bodies, and they just basically rely on what you eat, or they can make amino acids on their own, because not often are you going to have a balanced diet, or you're always going to give them what they want. So they're just floating around, and they're like, all right, if this guy's not going to eat what I need as far as certain amino acids, that I can then take a message from my DNA and create a protein, kind of taking all those amino acids and breaking them apart through hydrolysis and then resequencing them to make a protein that I need inside of me. I'm going to make it myself. And tryptophan is one of those amino acids. Here it is. You remember tryptophan? There's like this urban legend, but it's not a legend, that it makes you sleepy, right? It's the thing that's in turkey. So people say, oh, after Thanksgiving dinner, everyone falls asleep because there's a lot of tryptophan in this meat. And it's true. Tryptophan is a component of the B vitamin niacin. And then niacin is a component of serotonin and melatonin, which are kind of these wake sleep cycle regulating neurotransmitters that we have between the neurons in our brain. And so if you have a bunch of tryptophan, you might make more niacin, you might make more serotonin, you might get sleepier. You should try it. Well, most of the time, you're not eating tryptophan. And so you can see down there in the bottom left corner that we have an inactive repressor because tryptophan isn't binding to it and it's not repressing this tryptophan from being created. So if you look on the right side there, there's a gene and the RNA polymerase is going to go down it. And then it says down at the bottom right corner, polypeptide subunits that make up enzymes for tryptophan synthesis. So the bacteria is going to make its own tryptophan. It's like, I'm not waiting on you to eat turkey, so I'm going to make it. Tryptophan is absent, the repressor is inactive, and the operon is turned on. Let's make some tryptophan ourselves. We're not going to wait. Inactive repressor, no problem. Well, let's imagine that you have a big turkey dinner, and you then the bacteria is like, hey, I don't need to make tryptophan. It's in my environment. I'm just going to get it in here by diffusion. So if tryptophan is in the environment, it gets into the bacterial cell. It's going to bind to the repressor. You can see that there. It's going to change the shape of the repressor. It's then going to go on to the operator region of the DNA, and it's actually going to turn off its own production. Isn't that pretty cool? Tryptophan is present, the repressor is active, and the operon is off. Why should it make its own production if it's in the environment? You know what? I had this friend, my friend Vince. He would fall asleep all the time, drop of a hat, middle of the day, be sitting next to you talking, and then all of a sudden you'd look over and he was like snoring or sleeping. And I was thinking, if someone had a mutation in this gene that creates tryptophan, then what if you couldn't repress the production of tryptophan, and maybe you're making tryptophan all the time, which means you're making more niacin, which means you're making more serotonin, which means maybe you have a narcoleptic problem where you fall asleep all the time. Maybe this is the fundamental basis of narcolepsy. I have no idea.
Bacterium Escherichia coli, a group of 5 genes code for enzymes required to synthesize the amino acid tryptophan. All 5 genes are transcribed together as a unit called an operon. An operon is a group of genes that is under the control of a single operator site. A regulatory protein called a repressor can bind to the operator site and prevent transcription. When tryptophan is lacking in the environment, the repressor is inactive. RNA polymerase binds to the promoter site and then proceeds down the DNA, transcribing the genes for the tryptophan biosynthesis enzymes. When tryptophan is present in the environment, the organism no longer needs to make tryptophan. Tryptophan binds to the repressor and activates it. The activated repressor now binds to the operator located within the tryptophan promoter and blocks transcription. The tryptophan repressor is a helix-turn-helix regulatory protein. When tryptophan is absent from the environment, the repressor is in an inactive conformation and cannot bind to the DNA to prevent transcription. When tryptophan is abundant, 2 molecules of tryptophan bind to the repressor. This alters the orientation of the helix-turn-helix motifs in the repressor and causes their recognition helices to fit into adjacent major grooves of the DNA. Thus, the synthesis of tryptophan occurs when it is needed, but is repressed when tryptophan is available.
I hope that you're amazed by this. It's so complicated, and it all works because it's been subject to the pressures of natural selection for so long. Any mutation that was created in any DNA which created something that was a little bit different, maybe that helped this whole process of digestion of food or creation of these proteins to be better, or maybe it was worse. And if it was worse, that organism didn't fare as well in its environment. Maybe it didn't reproduce and pass along those genes. Maybe if it worked a little bit better, then it did pass along those genes, had more offspring, and over time that kind of DNA mutation became the norm. It's just fascinating how it all works. To me, I hope that you're amazed. I can help you more with this. I'm better at this stuff than chapters 14 and 15. Mr. Anderson can help you also. Go ahead and turn to your neighbor. Tell them what you know about the trip operon, please. Well, sorry about this picture. This is the best I could do about lactose intolerance. We'll get to lactose intolerance in a minute, but one of the sugars that exists out in our environment, sucrose, glucose, fructose, one of them is lactose, and people like having lactose, and it's this great source of, it's a sugar that gives us energy, and there's a lot of protein involved with dairy stuff as well, and so a lot of people really like to have dairy. If you notice here, you can see the sugar lactose in the bottom left corner, and you can see that lactase is the enzyme. And actually, I have it here in powdered form. There's lactase enzyme. I'll tell you why I have it in powdered form in a second. Lactase is an enzyme that a lot of people make, and that breaks lactose down into glucose and galactose. We use this as an energy source. Well, bacteria use this as well. You can imagine a E. coli bacteria floating around your large intestine, and it's like, sweet. If this individual takes in dairy, I'm going to break down that lactose sugar that diffuses into my single cell, and I'm going to use it as an energy source. But most of the time, you're not drinking milk, right? You're in class or whatever you're doing, unless you're drinking milk in class or whatever you're doing. You're not bringing dairy into the body. So the point is, what the bacteria does is it says, I'm not going to make the enzymes that equal lactase that are going to break down this lactose sugar, because there's no lactose sugar in the environment right now. And in that instance, you can see here with the left hand side yellow arrow, it says that the repressor is active, which means it's repressing the gene from expressing what it needs to create the proteins, to create the lactase that breaks down the lactose sugar. It just doesn't need to do it. And so no inducer is present to induce that repressor to get off there, and the gene is repressed. But on the right side, the lac operon is an inducible operon, usually off, and it contains genes that code for enzymes that are used in the breakdown of lactose milk sugar. But on the right side there, you can see that an inducer molecule has come and grabbed that repressor right off there, pulled it off, and now the genes can be expressed to create the lactase enzymes to break down the lactose sugar. See that on the right-hand side. The cool thing is that the very thing that creates the very thing that induces the creation of the enzymes that break it down is in fact lactose. Isn't that amazing? How efficient is that? It's like it comes in and because it's there, then the bacteria in our cells are going to create stuff to break it down. But if it's not there, then we won't create the stuff to break it down. And there's no message here. It's just the thing itself, the lactose sugar itself. It's amazing, I think. So that's the little green ball there that's stuck onto the red repressor. That is lactose sugar. It causes production of enzymes to break itself down. If I could fall right now, I would. I just think it's so amazing. So let's summarize this. So if lactose is absent, the repressor is active. It has a shape where it's able to bind onto the operator region, turn off the operator, turn off the gene, and no lactase is going to be created. Here's kind of what it looks like. There's the operator region. Imagine that's a huge long strand of DNA which goes above and below your computer screen. And then there's a repressor protein fitting in there onto those nucleotides. It's just amazing how it all works. But if the lactose is present, you can see that little green ball there. It's actually going to bind to the repressor, change its shape. Then the repressor can't bind to the operator region on the DNA. And then you're going to get those three things made way over there on the right side, those three things that equate to lactase enzyme. Well, some people are unable to make lactase enzyme, and so they're called lactose intolerant. They take in milk sugar, and it gives them gas and cramping, and they just don't feel good when they eat cheese or yogurt or things like that. And about 65% of the current world's population is lactose intolerant, including about 95% of the people in sub-Saharan Africa. And the reason why this is, I read, was because, well, let me tell you who makes lactase enzyme generally are Northern European descendants. And the reason why is because the cow was domesticated about 10,000 years ago, and apparently this ability to digest lactose by the creation of the lactase enzyme evolved independently in all these different pockets of groups throughout Europe during the thousands of years after that. So you can imagine that there was a selective pressure on a group of people. They had brought in cows, and cows were secreting milk, and people were drinking it. And they were like, wow, this is great, and look, I'm getting strong, and wow, I have more energy than everyone. And then other people in the tribe were like, wow, every time I drink that stuff, it gives me bad digestive system or whatever, and I can't really do it. And so you can imagine, and this is harsh to say, over time that the people who had this new protein and sugar source with this cow that was living outside of their hut, maybe they had more mates, maybe they reproduced and thrived more efficiently in their environment, and maybe they pass along those genes. And now today, lots of Northern Europeans are able to digest milk because of that situation. Well, here's what I'm going to ask you to do. If you are able to break down milk sugar, what I'd like you to do is go home tonight, and I'd like you to cut open your finger. And then once you cut open your finger, I want you to dip it into milk. So cut your finger, insert it into milk. And so the idea is that you have a cell here that it doesn't usually digest lactose or milk sugar, but it has the ability to, right? Because every cell has every gene, because they have all of the same DNA, but this one just has a repressor protein sitting on the operator region saying, don't create the lactase enzymes, because you've never needed them here, but now you do, because you're going to cut your finger, you're going to insert it into milk. Then what should happen is that the milk should bind onto the repressor, pull it off the operator, and you should make essentially lactase enzymes. Then, of course, what should you do? You should then get some other bit of dairy like yogurt or cheese and put it on the end of your finger. Just sit there, and if you watch, it will begin to break down over time, because the cells, although they don't ordinarily do this, you've induced the creation of lactase enzyme in these. I'm just kidding. Don't cut your finger. Cut someone else's. I'm just kidding. Okay, I'm just kidding. I'm kidding. I'm just trying to make an example here. But theoretically, I think it would be very interesting. I'll shut up now.
E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon: beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
If you need more, you can work with Mr. Anderson or myself. Go ahead and teach your neighbor about the lac operon right now, please. Finally, this is where we're going, genetic engineering, and we're going to talk about some of the ethical ramifications of that as we get towards the end of this chapter. Thanks, everyone, for your attention.